So you mentioned the paper that you've recently published and the idea of hyperfoods. You've mentioned that a couple of times. You're working on the DreamLab project that's funded by Vodafone, and you've just published this paper on hyperfoods. Just talk us through that very quickly.
Sure. I have to say that we've published it together and with your inputs. So it's a truly team effort, and the project that I'm currently leading with Vodafone Foundation, the idea is to use the processing power of thousands of smartphones to create a virtual supercomputer.
Yeah.
To crunch molecular data to identify new indications of existing drugs and find drug-like molecules in food that they have some disease-beating properties.
Mm-hmm. And what do we mean by disease-beating properties? How do you know that these ingredients have disease? What is it about them?
The way we did it in our manuscript, it's we've used the existing therapies and existing drugs actually to learn the molecular networks that are targeted by those molecules.
Yeah.
Because any disease process can be simplified to a process of interactions with the human molecular networks.
Okay.
That are responsible for different biological processes. And so we identified, so once we've analyzed the existing therapeutics in cancer using AI techniques that we use to simulate how the therapeutics affect human biological processes.
Mm-hmm.
And we learned what biological processes are targeted by existing therapeutics.
Okay.
And we were able, with classification
and by
accuracy,
therapeutics, we're talking about pharmaceutical drugs.
Yeah, we're talking about pharmaceutical drugs because they're designed to target disease processes. In cancer, it can be, for example, cell cycle, P453, for example, signaling processes, JAK-STAT pathways, and others. So there's a multitude of pathways. And once we've validated the model, we showed that it was able to accurately predict existing anti-cancer therapeutics based on the influence on molecular networks. So 9 out of 10 drugs were accurately predicted. And then we've took the model and processes and process thousands of known bioactive molecules in food to identify which one of them have the highest anti-cancer drug likeness. And we identify about 100.
So it's identifying the same molecules that you were looking for in the drugs and identifying them in foods.
It's not because they're coming from different molecular classes, but they target the same biological processes. So that's different because we didn't do it based on the chemical structure similarity, but rather than how they interact with biological processes. And we identified 110 molecules from different chemical classes that had the highest anti-cancer likeness, drug likeness. And then we, using multiple experimental databases, we map them into different food sources or different ingredients to identify which ones, from theoretical point of view. Theoretical point of view, I mean the number of different types of molecules that have cancer-beating properties have the highest cancer-beating potential. And as you mentioned, cabbage was one of them, where we identify about 9 different types of molecules that target different.
And now you drink cabbage smoothies.
And now every morning I drink cabbage smoothies. It's part of my it's part of my dietary plan. But also there were what's exciting that there was a whole diversity of molecules, and I would encourage people just to go and look at our food map because we actually clustered them according to their molecular composition as well.
Yeah.
And so ultimately, you need to study how all these molecules interact with each other to produce the effect of interest. There are a lot of limitations of the study because we have not taken the bioavailability of molecules into account, so they may not be present in high enough concentrations. That's why.
So we're saying that cabbage can have 9 different types of cancer-beating molecules, but it depends on where that cabbage is coming from, and it depends on each cabbage may have different levels of concentrations of those compounds in it, and depending, obviously, then what we were talking about earlier, how you cook it, how you process it, and so on, that will also be determine how effective those compounds are.
Yeah, I mean, this is the case because, for example, if you take the cabbage that is organically grown, I don't know, in California, and the one that grown, I don't know, in greenhouse in China, I mean, the chemical composition would be different. And as a result of that, the positive pro-health effects may be quite different.